This video will explore meiosis, which is a special type of nuclear division that occurs during the formation of reproductive cells, such as sperm and eggs. We'll also explore how meiosis serves as a source of genetic variation within populations. First of all, just a few introductory remarks about meiosis. Again, as we'll see, meiosis plays a big role in establishing genetic variation within populations, and we'll see how that occurs in just a bit. The new cells that are formed as a result of a cell dividing by meiosis contain half the number of chromosomes that you begin with, and this is a big difference between meiosis and mitosis. Also, again, meiosis plays a big role in sexual reproduction, whereas mitosis is involved in normal cell division in most parts of your body. The stages of meiosis are somewhat similar to mitosis, but there are some key differences. So, for example, we look at meiosis, it occurs in two distinct phases. So during meiosis I, what we'll see is that homologous chromosomes are separated. It's not until meiosis II that chromatids are pulled apart and separated, essentially just like what takes place during mitosis. Here we see those stages illustrated graphically. So if we start out with a diploid cell, perhaps not the best example because there are only two chromosomes. Nevertheless, this is a diploid cell because, again, there are two of each chromosome. During meiosis I, the homologous chromosomes are separated. Meiosis II, as we'll see, the stages are pretty much like what occurs in mitosis, but again, during meiosis II, the chromatids themselves are separated. So if we start with a diploid cell, the products of meiosis result in 4 haploid cells. Haploid because notice there's just one of each chromosome found within each cell. Also, I should point out that these 4 cells are genetically different from each other and from the cell that we start with. And again, we'll see how that occurs in just a bit. So let's now briefly summarize what occurs during each stage of meiosis. Beginning with interphase I. Interphase I is essentially the same thing as interphase of mitosis. So lots of things taking place. The key feature, of course, is that all the chromosomes replicate. So DNA synthesis, or the S phase of the cell cycle, occurs during interphase I. Prophase I, lots of things going on. One of the key features that occurs during prophase I is this process of crossing over. It's illustrated in this image here. So crossing over is essentially when 2 homologous chromosomes come together. That's what's illustrated toward the bottom of this image here. The two homologous chromosomes come together and they actually exchange pieces of DNA between each other. So alleles found on the maternal chromosome are exchanged with alleles on the paternal chromosome. So we have this reshuffling of alleles that occurs during prophase I of meiosis. Other events taking place too that are characteristic of prophase, namely the nuclear membrane is breaking down and the centrioles are making their way toward opposite ends of the cell. So illustrated in this figure here, let's begin right toward the middle. Here we see prophase. So nuclear membrane is totally degenerated by this point, crossing over occurring between pairs of homologous chromosomes, and again centrioles making their way toward opposite ends of the cell. After prophase I is complete, we then enter metaphase I. Metaphase I, notice we have chromosomes lining up at the middle of the cell. However, notice that we have pairs of chromosomes lining up, not individual chromosomes like we saw previously in mitosis. So metaphase I, pairs of homologous chromosomes line up at the middle of the cell, each chromosome attached to microtubules. Anaphase I, then what we see are the homologous chromosomes pulled toward opposite ends of the cell. Next comes telophase I, illustrated toward the far left here. So telophase I, what we have are 2 clusters of chromosomes, perhaps followed by cytokinesis, so 2 separate cells here. But notice by the end of telophase I, we have already reduced the number of chromosomes by half. So we began with a cell that had 4 chromosomes. By the end of meiosis I, we now have cells with just 2 chromosomes. So by the end of meiosis I, we've already produced haploid cells. And I should also point out that these cells are genetically different from each other, and of course genetically different from the cells that we start with. These cells can now proceed through meiosis II. There is an interphase 2 that occurs. However, during interphase 2, no DNA replication takes place, since if you look, the chromosomes already consist of 2 chromatids. So we now enter prophase 2. The rest of these stages are essentially just like what happens during mitosis. So prophase 2, nuclear membrane is gone, centrioles moving apart toward opposite ends of the cell, metaphase 2, chromosomes lining up at the middle of the cell, anaphase 2, chromatids are separated, pulled toward opposite ends of the cell, and we end up with a grand total of 4 cells produced from meiosis. Each one, again, by the end of meiosis II is haploid, because notice each cell in this case only has 2 chromosomes, whereas we started out with a cell with 4 chromosomes. And ultimately, these 4 cells are genetically different from each other. So, to summarize a few key points, by the end of telophase 2 and cytokinesis, we form a total of 4 haploid nuclei, or cells, if indeed cell division takes place, and the new cells are genetically different from each other. If we consider this basic question, what causes genetic variation in populations? As we'll see, meiosis plays a huge role in this scenario. First of all, we've already discussed crossing over, this exchange of alleles between homologous chromosomes that occurs during prophase 1 of meiosis. But even if crossing over did not take place, we would still have genetic variation in the cells produced as a result of meiosis. How is that? Well, the second point we'll mention here is what's referred to as the independent assortment of chromosomes. Take a look at this image here. We're looking at a diploid cell toward the top. Again, 2 of each chromosome. These homologous chromosomes can line up during metaphase 1, much like we see here. If they line up this way, if you then run this cell through meiosis, what you'll see is that you'll form 2 different allele combinations. What we see here on the left, and this one over here on the right. However, it's equally possible that during metaphase, instead of the chromosomes lining up like we see here, they could also line up like we see here. So essentially it's the same chromosomes. These top 2 homologous chromosomes are the same as what we see on the left. These bottom 2 are flipped. Again, the way these chromosomes line up during metaphase 1, totally random. If they happen to line up this way, as we see on the right, what we see are 2 different allele combinations in the cells that are produced. So again, even if crossing over doesn't take place, we still get genetic variation in the cells produced from meiosis, again, as a result of this independent assortment of chromosomes. Again, it just comes down to the random way in which chromosomes line up during metaphase 1 of meiosis. So if you think about this for just a minute, we can ask the basic question, well, how many combinations are possible? Again, let's just ignore crossing over and pretend like it doesn't even take place. Just based on independent assortment of chromosomes, simple formula that you can use is 2 to the nth power, where n refers to the haploid number of chromosomes. So if we consider our previous example, the diploid cell that we begin with contains 4 chromosomes. The haploid number of chromosomes would therefore be 2. So 2 to the nth power, 2 times 2 gives us 4 different combinations of chromosomes. And again, that's what we see illustrated here. Also, keep in mind that we have crossing over taking place, we have independent assortment of chromosomes, and we have these events taking place in males and females. And of course, males and females have to come together in order for sexual reproduction to occur. So by having the formation of gametes by meiosis in 2 separate sexes, this too compounds the genetic variation that we find within populations of organisms.